Going to see about the viral genome. Viral genome refers to the genome to be present inside the virus. Virus is found to be a organism, a microorganism that is going to be a living microorganism inside the host, and in the absence of host or outside the host, it can be a functional as virions, but outside the host it can be a non-functional too. Inside the living organism, inside the host it became a living organism because it can undergoes the replication process and increases its number inside the host by hijacking the protein machinery of a host. Or the host can be a plant or animals, or it can be a bacteria. The host changes based on the virus causes infection. Some virus may infect plant, they are known as plant viruses. Some infect animals, they have animal viruses. And the virus that going to infect the bacteria are known as bacteriophages. So now we are going to see about the virus genome. A viral genome is going to be present in virus. A virus, before that we should know what is virus. Virus is going to be a small infectious agent that can infect a plant or animal or a bacteria. It can infect any of this organism. It infects by infecting the organism. It can replicate inside the organism. That means it can replicate inside the living cells of other organism where it's going to infect. Therefore, the viral particles or virions, or viral particles or virions are going to be a functional virus that is found outside the host. In general, a virus is going to be consists of three elements. They are a genetic material that can be a DNA or a RNA. Then a protein coat that is going to be a capsid that is going to cover the genetic material. The outer covering is known as capsid, and the lipid envelope is going to be a inner covering. It is also going to cover the genetic material that is found to be inside. Now we are going to see about what is genome, viral genome. A viral genome can be a DNA or RNA genetic material. A DNA viruses are going to be a viruses with DNA as a genetic material. A DNA of these viruses can be a circular or linear. Sometimes it can be a double-stranded or a single-stranded too. RNA viruses are the viruses with RNA as a genetic material. They are going to be a double-stranded or single-stranded. Most of the RNA viruses are going to be single-stranded. It can be circular or a linear. A genome size of RNA viruses can be varies because it's going to be a single-stranded. Size also varies. The RNA viruses has a smaller genome size than the DNA viruses. Most of the DNA viruses are going to be double-stranded and RNA viruses are going to be single-stranded. DNA viruses are going to be double-stranded, most of them, and most of the RNA viruses are going to be single-stranded as most of the prevalent to be found. RNA viruses are going to have a smaller genome size than the DNA viruses. DNA viruses are going to be, most of them are double-stranded, going to have a more genome size than the RNA viruses. Next, we are going to see about the bacteriophage. Bacteriophage is going to a virus that can infect the bacteria is known as bacteriophage. A virus that going to infect the bacteria are known as bacteriophage. It can be either lytic, it can undergo replication either by lytic or non-lytic cycle. It replicates within the bacteria. As it's going to infect the bacteria and it can replicate inside the bacteria. It is going to be encapsulated by the protein coat that is capsid. A genome that is going to encode for the bacteriophage may be 4 to 100 S of genes in number. First, we are going to see about the M13 bacteriophage. M13 bacteriophage is a virus that is going to infect the E. coli. E. coli is a bacteria, a common bacteria to be present. It was infected by the M13 bacteriophage, but it undergoes a non-lytic cycle. It composed of single-stranded DNA. It's going to consist of a DNA that is single-stranded, and it has a protein coat that is going to be present as a tube-like structure of capsid protein to be found outside the single-stranded DNA. In the single-stranded DNA, it's going to consist of 6,407 nucleotides, which is going to encode the 10 genes that is going to encode the protein, and one gene must be an intergenomic sequence. Among these 6,407, they are going to be 11 genes. Among them, a 10 gene is going to encode a protein, and the one gene is going to has a intergenomic sequences. This is a M13 bacteriophage structure. Here it's going to be a ORI. ORI means origin of replication. It is a region where the replication is going to be started off. G2P, replication endonuclease. It is a new endonuclease. Replication endonuclease going to create a nick in the double stranded. It's going to be a single stranded but became double stranded during the replication. Therefore, it creates a nick at the region of double stranded. Then it's going to be adsorption protein. Adsorption protein is going to be very important for the adsorption into the host, that is bacteria. The phage get adsorpted into the bacteria through adsorption protein. Capsid protein is going to be a covering above the outside the genome. It's going to be synthesized by G6P genome sequences, where adsorption protein is going to be synthesized by G3P genomic sequences. Then G1P is going to be morphogenesis. Morphogenesis means assembly of virus elements. A virus elements is going to be capsid, genetic material. These are all get assembled by this morphogenesis process. This assembly is going to be carried out by this genome, a protein synthesis by this genome. Here we can see the G3P is going to be at this genome. This genome is going to synthesize the adsorption protein. This adsorption protein is very important for pilus mediated adsorption. That means it is a E. coli bacteria. A phage can infect this bacteria by injecting its genetic material into it. To infect its bacteria, it must get adsorbed to the E. coli. For the adsorption, it needs a certain protein, that is adsorption protein. This adsorption protein is going to be synthesized by G3P gene. Therefore, pilus mediated adsorption from virus to the host cell is going to be carried out by this adsorption protein synthesis by G3P gene. Then G6P, G7P, G8P and G9P are the capsid protein synthesizing genes. This capsid protein is going to be very important for the injecting of viral DNA into a bacterial membrane, and also it provides a protection to the genetic material. And next, it's going to be G2P gene. It is very important gene to carried out the replication. It is going to involve in starting the replication. We are going to see the replication in the next slide, so that here the replication is going to be started out by these replication endonuclease. Therefore, replication endonuclease is very important. This replication endonuclease enzyme is going to be, or a factor is going to be synthesized by this G2P gene in the M13 genome. So M13 is going to be carried out the rolling circle model of replication. For that, we should know what is rolling circle model of replication. The rolling circle model of replication is going to carried out in the single stranded DNA in the positive strand, especially in the positive strand viruses such as M13 or T4 bacteriophage. For that, here we can see the single stranded DNA of the M13 bacteriophage. As we already saw that bacteriophage of M13 is going to be single stranded, which is going to be covered by the capsid protein as tube-like structure. So the single stranded capsid protein is going to be circular in manner, a circular single stranded genome is covered by the capsid protein. So single stranded genome is, before that we should know that E. coli is going to be infected by the bacteriophage. It's going to inject its genetic material. It is a M13 phage. So its genetic material is going to be single stranded circular. After infect, this genome is this. This is a genome to be injected into the E. coli. After it enters into the E. coli, this is the steps is going to be taken place for the replication. For that, for the starting of replication, this single stranded positive stranded DNA is going to create its negative strand. Therefore, it becomes double stranded DNA at first. Then, as we said before, the genome G2 gene is going to synthesize the replication endonuclease. An enzyme or a factor is going to be synthesized by this genome after it became double stranded. This replication endonuclease is going to create a nick in this double strand in the positive strand, that is a phage. Therefore, a negative strand is going to be intact in it, but a positive strand is going to be created a nick by replication endonuclease synthesis by G2 genome. So, after that, this 5' region is going to be attached to the plasma membrane of a E. coli. This is the plasma membrane of a E. coli, and the 3' end is going to be free. So, after that, from this 3' end, a 3' end of a gene is going to consist of 3'OH. Therefore, the 3'OH is a free end where the nucleotides can be added. So, the nucleotides in the E. coli is going to be added here. From the E. coli, the nucleotides are gathered and added into this 3' end in the presence of negative strand, a replica of a positive strand, that is negative strand. A complementary is going to be synthesized from the 3' end. Therefore, it is a newly formed strand. As it moves on to synthesis newly, it rolls back and the DNA is going to be relieved of. So this is a negative strand. A new strand is going to be synthesized here. As it moves on to synthesis the new strand, this DNA, that is positive strand, is going to be rolled back. Therefore, it is known as rolling circle model of replication. As it was rolled back, it is known as rolling circle model of replication. As this structure is going to be like a inverted sigma structure. Therefore, it is known as sigma replication 2. As we see before, the bidirectional replication, where it's going to be like a theta-like structure, therefore it is known as theta replication 2, like that. This structure, due to rolling back, and in this replication is going to be similar like a inverted Greek letter sigma, it is known as sigma replication. Therefore, where there is a replication is going to be taken place from the 3' in the form of leading strand, whereas a plasma membrane attached to the 5' end. From this 5' end, from here there is a continuous synthesis of RNA primer is added, and a continuous synthesis of lagging strand is going to be synthesis from this 5' end in the discontinuous manner in the tail region. Therefore, after rolling back of negative strand, a replica of a positive strand, a full DNA is going to be synthesized after it reaches the origin. That means a region where the replication is going to be started off. When it reaches the origin, it going to stop its replication. A continuous leading strand has been synthesized in the tail region and its lagging strand is synthesized. Therefore, a progeny DNA is going to be synthesized. One is going to be a newly formed DNA and another is going to be a replica of positive strand, that is so that it became circularized and lead to formation of two strands of daughter virions. Therefore, it is a two strand. So here we can found that this is a newly formed strand, that is positive strand, and this is a newly formed lagging strand, that is going to be a negative strand, which is going to be involved in next replication. A newly formed negative strand is going to be involved in next replication process for the positive strand synthesis. This is a newly formed positive strand which is going to be assembled into the capsid and lead to formation of phage. After these replication ended off, it is assembled into the capsid. From the capsid, it lead to formation of daughter phages, daughter M13 phages. After the daughter M13 phages is formed, it gets released from the E. coli by budding without lysis. Therefore, it is known as non-lytic cycle. In the image, a replication, rolling circle model of replication is also known as sigma replication because its replication, during the replication, the DNA structure is going to be similar like a sigma, inverted sigma, Greek letter sigma. Therefore, it is known as sigma replication. A rolling circle model of replication is going to be present in the single-stranded DNA of a T4 phage or M13 phage. A chromosome first became double-stranded when it enters into the E. coli host. It became double-stranded where the positive strand, that is a original strand, is going to synthesize a negative strand and create a double-stranded. Then it's going to create a nick at the positive strand, that is original strand is nicked by endonuclease. An endonuclease enzyme is going to create a nick in the positive strand. As a result of cut, a 3-OH group, a 3-OH group is going to be a growing end. Therefore, nucleotides are going to be added, that is complementary to the 5' end, complementary to the circular unbroken circular negative strand. Therefore, during the nick formation, the 3' end is free to rotate. The unbroken end is 5' end is going to be attached to the plasma membrane of E. coli, that is host bacterium is E. coli plasma membrane get attached to the 5' end. And in the 5' end too, a lagging strand is going to be synthesized, but in the 3' end, it going to synthesize the leading strand, where it is complementary to the negative strand. The broken strand is going to be attached to the plasma membrane, that is going to serve as a tail region and becomes a template for the lagging strand. The unbroken strand acts as a template. From that, a complementary strand of positive strand is synthesized. Therefore, as the circular strand is going to be rolled back, it is known as rolling circle model of replication. As it rolls back further, it gets unwinds. It unwinds and synthesizes the leading strand till it reaches the origin, where in the case of lagging strand to be synthesized in the 5' end, it lead to synthesis of RNA primer is going to be added in every lagging strand and lead to synthesis of nucleotides at the 5' end. But in the 3' end, it is going to be continuous manner as leading strand is going to be synthesized, as only one RNA primer is going to be added off. After completion of the synthesis, a tail is cut off from the plasma membrane by the endonuclease, which creates a nick and lead to production of progeny rod. As we saw in the structure, there is a progeny rod that is going to consist of one newly synthesized DNA and another is going to be a positive strand DNA. The progeny rod is going to undergo circulation, which lead to new circular molecule. The gap is closed by leakase enzyme. Therefore, that's what a rolling circle model of replication lead to newly formed circular molecule. This is a structure to be explained.